My name is Breann Brown. I'm an assistant professor in the Department of Biochemistry. The focus of my research involves looking at mechanisms of protein assembly that's involved in mitochondrial physiology. And the reason we're interested in that is because there's a growing body of evidence that shows there's a large number of people that have some sort of mitochondrial genetic-based disease. And so we're interested in understanding how proteins assemble and then how these assemblies are no longer functional in certain diseases. And some of the diseases that are affected include various blood disorders, as well as some early onset neurodegenerative diseases. So what attracted me to my area of research is that I've always been involved in how proteins come together to mediate cell signaling. Now we just do it in the focus of looking at mitochondrial physiology and signaling. So we know that there's a large number of mutations that can lead to human disease, and so we're interested in looking at how these mutations have a structural consequence. And so what drew me to my area of research was taking this information that we know from patients and kind of boiling it down to a very fine, detailed level so that we can then begin to figure out better ways to treat these diseases. Basic science is important to my field of study because in order to understand how these diseases are manifest in humans, we first need to understand how the framework and the groundwork for how proteins are affected. And so when you have alterations in protein structure and function, that then leads to alterations in cell signaling and so on and so forth. But to begin to understand how we can develop better treatment strategies, we first need to lay that groundwork to figure out how the system works properly and how when you have mutations, how there are changes in the system that then lead to disease. So funding is critical to carrying out our research, not just for purchasing equipment and supplies and reagents, but it's also critical for funding future generations of scientists. And so a large part of what we do is train students and other postdocs and technicians in science, and so we need funding to support that training, both at the bench and in their careers. So a diversity of funding is very important for our research. The main things that attracted me to Vanderbilt were, one, the research resources, the access to core facilities and state-of-the-art instrumentation so that I could do all of the experiments that I need to do now and that I can think to do in the future, but also the community at Vanderbilt attracted me. In the Department of Biochemistry and across basic sciences, I've met very helpful individuals and people who are open to collaboration, and it's really enabled my lab to get up and running very quickly. One of the unique features about Vanderbilt that attracted me here was a range of scientists at different stages of their career who are all working together. So at the junior level and at the more senior level, there's great communication and there's great support and mentorship available here that provides an atmosphere where scientists of all career stages can be successful. So advice I would have for students and postdocs who would like to enter this field is, one, be persistent, because science is not linear and there's often deviations, and so you have to push through those deviations. Also, be curious. Be able to ask your own questions and figure out how you can pursue those answers. And then also I would say you have to be meticulous. You have to take good notes, and you have to be able to know what you did in the past, know what other people have done, and how can you improve and learn from those previous experiences. So I did my graduate work at Brown University with Professor Rebecca Page, which is where I learned my structural biology expertise. And then I moved to MIT to work with Professor Tanya Baker in the Department of Biology for my postdoctoral research, where I expanded on this expertise and then also learned some new techniques that we're using here in the lab at Vanderbilt.